So I hope you can all hear my voice.
Yes, ma'am.
Okay, this is good. Okay, Zaraksha, can you give me this information that how many of you are, excuse me, how many of you are actually taking industrial microbiology? Are like your second semester, your all of second semester is the entire second semester is taking industrial microbiology. So are you 15? How many students are in second semester right now?
Ma'am, I think 20 plus.
There are more than 20. Okay.
Yes.
Okay. So according to that, our participation is less, right? I mean, so like in
Ma'am, due to weather signal issue.
due to signal issues. Yeah, we've been having this signal and internet issue throughout our this spring session as well. So how was your spring session?
It was good.
You did cover it online, right?
Yes, ma'am.
Mm-hmm. So how was it?
It was good
It was good.
almost.
Almost okay. All right. So, okay, let's continue where we left. So, we were talking about importance of recombinant DNA technology and its importance. I mean, recombinant DNA technology has influenced um, a lot of industries, all of them, which includes biology, one way or the other, scientific advancement, or whatever it was. So obviously, it came to microbiology as well, and it came to industrial microbiology as well. So we covered this part. Let's move on. So what we're trying to, it's now giving us an example of how microorganisms that are actually giving us uh, that link between a recombinant DNA technology and microorganisms, right? So this slide over here is talking about that because when you're trying to express, when you have expression vectors, they are basically in bacterium, and one of the model bacteria that is being used for hosts and for expression of products and proteins is Escherichia coli, right? So, as with some human proteins, there are eukaryotic host, which is also being used, which is a yeast, right? So there are two kind of host organisms and expression of proteins which is being used, and both of them are they fall in umbrella of microbiology. It's either the bacteria, or it's either the yeast, or it's either the fungi. So basically, there are few shortcomings of using Escherichia coli, of using E. coli. There are certain kind of post-translational modification that does not happen in bacterial hosts. So you shift towards an eukaryotic host. You shift towards using a fungi and a yeast so that you can express different elements. So this is just an extension of our um, um, previous slide where we were talking about using recombinant DNA technology as a benefit, adding benefit to the entire industrial microbiology, right? So now there are advantages of fermentation. Let's look into that. Since the entire process of industrial microbiology, it revolves around fermentation process itself. So it's good to look into certain advantages and a vast range of important products, many of which were formerly manufactured by chemical processes are now most economically produced by microbial fermentation. Now, most economically produced, meaning now there is less cost, so it is more cost effective. So there is less expenses that are being put in. So what was being developed with chemical process is now more efficiently being developed using microbial fermentation, right? And also biotransformation processes. So this biotransformation process is where you change the product chemically to a different product. So you actually doing transformation of chemical products, but you're using the enzymatic system present within the microbial cell, right? So there are different kind of transformation processes you can do, and one of the transformation processes that we can do is there are um, pharmaceutically produced hormones, right? For different kind of endocrinological ailments, there one of the treatments is to provide the hormones in form of tablets, to provide the hormones externally through oral consumption, right? So biotransformation is most commonly used in pharmaceutical industry, where you want to convert a main chemical structure into different derivative structures. So basically, you're doing transformation of that main organic skeleton, right? So all of this process can happen in the end. You can get many derivatives of the products, right? So microorganisms also provide valuable services. They have proved to be particularly useful because of the ease of their mass cultivation, because of speed of growth, use of cheap substrates that in many cases are waste, and diversity of the products that you get, right? So this is kind of summing up our six main advantages. And one of those six main advantages is that microorganisms are very easy to grow, right? So if you want to grow them in very large quantities, which is required in the process of fermentation, then it is possible, it is economically possible, it is cheap, and it is easy to cultivate them. It is easy to achieve a very speedy growth as well, number two. Number three is that the kind of substrates and the raw materials that these microorganisms they consume during their growth and during their cultivation is also something very cheap. So the raw materials that is going in and the money that is going in to set up this fermentation, it also is very cost effective. And then in many cases, you can use waste, right? So in order to have a good cultivation media, you don't need very special, refined, molecular grade products, very pure products to go into fermentation. In fact, you can use several wastes which can be consumed by microorganisms. So you also have this sustainable and environment friendly process in your hand. Then, in addition, their ability to readily undergo genetic manipulation has almost opened some sort of limitless possibilities. So, of course, like we just looked into this last point here, that addition of their ability to undergo genetic manipulation, this is this is the same point we were studying just about recombinant DNA technology, that now you can change the genes, you can change and you can diversify the entire organism, and you can diversify their activities. And if you can do that, the entire fermentation product and will change. So the services and products and magnitude and their effect on different industries would simply change. So these are some few, but actually very major advantages of fermentation, right? So it looks like something very brief, but we have kind of looked into some eight dimensions of advantages of fermentation. It kind of includes eight important points, which are there as advantage of fermentation. Now, what we're going to look into, we're going to look into a typical framework of an industrial process, right? So there is this microbiology part and the science of microorganism and knowing the property of microorganism, how to grow them. And then there's this typical industrial process, right? So when it comes to establishing industries, it means that there has to be some special kind of SOPs, certain kind of specific procedures that you have to follow to ensure that everything happens the way they are is smoothly, and management of these processes is simple. Whoever is running these processes, for them it's simple and easy as well. So basically, successful development of fermentation process requires major contribution from a wide range of disciplines, right? So what they're trying to say that when you are establishing a industry and in which you are employing fermentation, so microbiology is of course the main field that is making certain contribution, because you're using microorganisms, their traits, their characteristics. But then there are other disciplines which are playing a very vital role in the entire process. And what is that? There is involvement of biochemistry, because there is certain kind of, like we just said, that there are biotransformations that also take place. So biotransformation process has a lot to do with the biochemistry and organic change of the structure that is taking place. So that that is something very important. Then if you are living in this, we're living in this modern era, and most of the strains, they have been genetically modified. So without the knowledge of genetics and molecular biology, so alone the knowledge of microbiology would not do much. You need genetics contribution, you need that knowledge of molecular biology, chemistry, chemical processes, and also mathematics and computer technology. Why? Because most of the industrial processes, they have been automated. So if the entire processes have been automated, then you have to, you need mechanical engineering. Mechanical engineering and material engineering is very important. So how you put in a fermenter, how you apply the fermenter, and when you automate the fermenter, like most industries are, there are few industries which are manually operated, and then there are few industrial processes, and the entire things that happen, they have been automated. So in order to make it automated, of course you need this computer technology to play a role. So you can see when when you enter into industrial knowledge includes a wide range of disciplines. So basically, the entire process, industrial processes and operation, is divided into two stages of any industrial process. One is an upstream process, upstream processing, which you call USP, and the other is downstream processing, which is DSC, right? So we just abbreviate them. But it's just an acronym, right? Upstream processes, and then downstream processes, DSP. So what is USP? What is an upstream processing? Upstream processing is associated with all factors and processes leading to and including the fermentation, consisting of the three main areas. And in the next slide, we're going to look into those areas. So what is the upstream process? Upstream process is um, uh, all the preparation, all right, all the factors, element, and the process which is included in order to set up fermentation, right? So fermentation itself, which is taking place, meaning the media and the microorganism and the fermenter and the vessel in which you set up that fermentation, all of that, and everything and all the preparation process to make that fermentation happen. That is your upstream processing, right? So it consists of the three main areas. Let's look into what these three main areas are. So in an upstream process, area number one is the producer microorganism, right? So what will perform the fermentation process? Of course, it is the microorganism. So that main microorganism, the number one thing that is part of the upstream process is having that microorganism, the primary inoculum, which is going to perform the entire fermentation process, right? So the strategy for initially, it might include other steps, right? How you come up with this microorganism. So whatever an industry or the person who are working on this, whatever processes are required to get you the producer microorganism through which you're going to set up that fermentation, all of this is included in the upstream process. So it includes the strategy for initially obtaining the suitable industrial microorganism. So how you're going to screen the microorganism, how are you planning to obtain that microorganism that will perform the fermentation. Of course, all that strategy is part of this producer microorganism. Then there is strain improvement to enhance productivity and yield. So all this process is also there, meaning if you have a microorganism, but it's not doing the job you want to achieve from it, you might go towards enhancing the productivity and yield, and you can do it by recombinant DNA technology, meaning you can improve the existing strain. So if there is this research process which are involved in improving the productivity and the yield, all that research process and experimentation which is going on is part of the upstream processes, and also maintenance of that pure strain purity. So once you have that desired strain, and once you have that microorganism that is giving you the kind of product, and it is good with productivity and the yield and all the traits and qualities you would want your fermentation and primary and producer microorganism to have, you would want to maintain that. So whatever experimentation and lab work which is required to maintain the purity, to maintain the pure culture, it is the process of USP, right? It is an upstream process. Then preparation of a reliable inoculum and continuing development of selected strains to improve the economic efficiency of the process. Again, how you prepare that inoculum, and then how do you continue to enhance that strain further. All of these are included in producer microorganism and the upstream processes. So, for example, the production of a stable mutant strains that vastly overproduce the target compound is often essential, right? So, the example written over here that the natural strain of the bacteria or the yeast, whatever you're using to make a product, it doesn't give you a very high amount of the product. So you naturally want a kind of a strain that is high on productivity, right? So what you're doing, you are inducing a mutant strain, and that mutant strain is so designed so that I can give you a higher and higher yield of the product, right? So the target compound is overproduced, right? So this high amount of productivity is basically required. So the production of the mutant strain and the kind of trait you want to induce is overproduction of the compound is required, and all of this is a kind of upstream process. Now, area number two, the other main area of the upstream process is the fermentation medium. So once you have the microorganism, you must have some medium in which that microorganism can grow, right? So there, the medium. What is medium? Medium provides those energy sources, the carbon sources, the thing which the microorganism takes nutrients from and is essential for its growth, right? So from where do you get those medium and selection of materials that you do and bring about that carbon and energy sources for this fermentation medium, right? So what are these? So you want to do in your upstream processing a cost effective medium and raw materials employment. So it includes essential nutrients, and it also includes along with it a media optimization with all the vital aspects, so that it can maximize the yield and the profit. So we are at this stage at a very early point. Right now we're in introductory chapter. Later on, when we're going to study these fermentation process, and we study the media and the kind of raw material that goes inside, we're going to see that there's so many aspects of the kind of material, raw material that you're using as substrates, which can actually minimize the yield. And if there is minimization of the yield, that also means you are low on the profit as well. Of course, eventually you're producing an industrial product, and that industrial product has to go into the market, and that if it goes in the market is definitely supposed to earn profit, right? So if there is low yield, it means low profit. So you do not want a media that is cost effective, but eventually it is also minimizing the yield. So what kind of processes and raw material you're going to use, what kind of fine tuning of the elements you're going to do, so that you get the cost effective media along with the maximum yield and the maximum profit eventually, right? So in many instances, the basis of industrial media are waste products and other industrial processes. Of course, when you're using a waste products for a fermentation media, of course, you are working on a cost-effective method. You're using a waste, which actually comes for a very low price, right? So you can go for sugar processing waste, you can go for cellulosic, lignocellulosic waste, which comes from the paper industry. You can go for cheese and corn liquor, um, which is actually a waste from many wine brewing and beer brewing processes, and also malt brewing processes. So it produces many of the byproducts which are actually waste for that industry, but can be useful as a raw material of fermentation in other industries. So usually these kind of fermentation media, they come from industrial wastes, right? Then another very important aspect of an upstream process is the fermentation process itself. Of course, the industrial microorganisms are normally cultivated under rigorously controlled conditions developed to optimize the growth of the microorganism or production of the target microbial product. So, what is this? Okay, so what is this fermentation itself? It's like having that media, and you put in into that media, you put in your microorganism, and you let it grow to the maximum, right? So this is the entire fermentation process, and you let microorganism do its stuff, do its biochemistry, do its transformation, whatever it has to do. So eventually, you have to set up a fermentation in such a state that your product is not minimized. Your product has to be maximized. Whatever you do, it has to be on the maximum, right? So the synthesis of microbial metabolites is usually tightly regulated by the microbial cells, of course. So in order to obtain high yield, the environmental condition that trigger these mechanisms has to be on the optimum, right? So you have to give the microorganism the kind of environment it takes to give you the maximum yield. So, whatever environment is suitable for the microorganism that results in a higher yield is desirable in this fermentation process. So, particularly repression and feedback inhibition must be avoided. So, what is this repression and feedback inhibition? Usually, you must have heard in biochemistry and other kind of processes as well that there is a process that is happening, and there is this biochemical reaction going on, and in the end of that biochemical reaction, when a product reaches its particular amount, let's say it reaches about 90% of the compound is made, that 90% compound itself acts as a inhibitor, right? So there are regulatory feedback and repression pathways which control the product. So it's like there is this cellular process, and when there is lack of a product, there is a pathway which begins to produce a product. But once that product reaches a high amount, the cell senses that high amount, and then it stops the entire regulatory process. But in terms of fermentation and in terms of production of high yield of product, you do not want this repression and inhibition to happen, which happens normally inside a cell, right? We have already seen these kind of regulatory pathways where one of the product also acts as a repressor. It's going to now shut down the process because enough of the product is around in the environment. So we do not want that in the industrial process anymore. We do not want the product we're after, it increases in such high number that the cell which is programmed to repress it starts to repress the entire process. So we are trying to regulate the fermentation process in such a way so that there is no inhibition and no repression of the process going on, right? So, and that varies from product to product, and usually it includes that what industries and do that they extract that product. They set up fermentation for some time and then they would extract their product so that within the media they do not have a high amount of that product, which would cause a inhibition of the process eventually, right? So what we've seen, we've been doing the three main areas which are included in the upstream process. Area number one was the producer microorganism. Area number two was fermentation media, and area number three was the fermentation, right? So how does this fermentation basically take place? Fermentation takes place within the fermenter, right? And then all the requirements of fermenter is also part of the upstream process, all right? Do include that requirement of fermenter is part of this upstream process. And this area number three, basically. When you're working on fermentation, you're working on the kind and the nature of the fermenter itself as well. So fermentation is performed in large fermenters, which are really large vessels, right? And in those vessels and tanks, there are different kind of controls. There are simple tanks, or there are tanks which are equipped with a stirrer, so they can be stirred, and then they are unstirred, and then they are aerated, unaerated. There are different forms of vessels and tanks that you have for fermentation to carry out. And fermentations, since they are in very large size, they have a very upscale size. It can range to thousands and thousands and thousands of volume, right? So your cubic measurement, your volume, it increases so much in size that in order to provide these fermenter with the required media, the water, the energy source, the inoculum, there must be these heavy pipe works that are going on. So the constructed material that is there, it should be stainless. There should be some pipe work It should be sterilized. Of course, we are producing kind of beverages, some kind of products, and once we're done producing that product, there has to be some sterilization process going on in those fermenters. So it's actually, we're talking, when we're talking about the fermenter, we're talking about the equipment. So it's very natural that if you're working in any, if industry is working on any kind of processes, they must have their sterilization process there. And then there are different kind of operations, mode of operations for these fermenters. It could be batch, fed batch, and continuous system. It could be aerated. It could be agitation. It could be different other variation. And by the way, we are going to cover all of this later on in our course as well. So now we come to downstream processes. So downstream processes are all unit processes that follows the fermentation, right? So what happened in upstream process, that we manage the fermentation, we manage the fermentation media, and we also looked into the equipment and the requirement of the equipment before we could set up the fermentation. So up till fermentation, it was upstream process. Now everything which comes or follows the fermentation process is the downstream processes. And of course, the fermentation is all that is taking place inside those tanks and inside those vessels. But what do we do afterwards? Of course, we have to extract the product. Without having the product in hand, the entire industrial process is of no use. So every process, there are a lot of processes, and actually the downstream processes are more critical in the entire industrial process. So what does it involve? And it involves cell harvesting, it involves cell disruption, it involves product purification. It also include how you're going to obtain the cell extracts and the growth medium, how you're going to purify the product from all of these growth medium. And of course, once you have the product in hand, there has to be many finishing steps in order that you get that final product which is ready to be packaged and put into market, or whatever use you have for that product. So there are a lot of downstream processes that are going on, and industrially these downstream processes are more critical and very important. So, however, attempts are now being made to integrate fermentation with DSP operation, which often decreases process product, often increases process productivity. So this very last point, what it's trying to emphasize is that the kind of fermentation you set up, I mean, the kind of upstream processes you adopt, there can be many, right? So the kind of SOPs you adopt to operate your upstream processes are supposed to be in line with the downstream processes, because if the extraction process from the from fermentation medium is too, I mean, the cost of extraction, the level of hard work, lab work, or the labor which is involved is too much, then your upstream process is not supporting your downstream process. So the entire process has to be, the upstream process has to be designed so that it supports the downstream process. So what is trying to say that we have to integrate our fermentation in such a way so that our downstream process operations are more productive. The entire process is more productive. So overall, DSP must employ rapid, efficient methods for purification of the product and maintaining it in a stable form. So main thing of the downstream process is how are you going to purify the product? All right. So once you have the product in your hand, how are you going to keep that product as stable as possible, right? So of course, if you have a product and if you have a good industrial processes going on, then you will remove any kind of impurities from those products, right? So in some products, especially enzymes, retention of their biological activity is vital. Since we've already seen that if you are using fermentation and you're using fermentation to produce enzymes, which are catalysts, and you already know the use of having enzymes in industry. So you can see how critical it is, because enzymes, they lose their activities. Enzymes, they lose their biological activities. They have to be kept in a very stable form. So if you have these products and you have these enzymes which are being produced and they lose their biological activity, then the entire process that you have done is of waste, right? It has went into vain. So you want to keep the product stable. So when it comes to enzyme, for example, you have to keep their biological activity in mind. And when you keep an enzyme stable, you're basically keeping its biological activity stable, right? So finally, there must be some safe and inexpensive disposal of the waste product as well. So within the downstream process, what how you dealing with the waste is also important, right? So you have set up a fermentation process, and there's a lot of things and a lot of thought process and a lot of materials that were into doing the upstream processes as well as downstream processes. There are a lot of work, and now you have in return certain waste from the entire process. So one of the important points of your downstream process also includes how you deal with the waste. So what this is the outline of the entire fermentation process. So you can see an upstream process here, and you can see a downstream process here. So what this upstream process includes, it includes all the fermentation raw materials. It includes production microorganism that we've seen, the producer microorganism, and the fermentation that you set up. So up till fermentation, and all the things that are required for setting up the fermentation is the upstream process. And what includes in the downstream processing is the product purification, obtaining, extracting the product, and developing that product into the form in which it is required, a stable, biologically stable product that you get in the end. And then effluent waste, right? So the effluent waste is also something very important. Like we said, how you deal with the waste, what you're going to do with the waste, how is this waste disposed of, is also part of a good practice. So it is also part of the downstream process. So this figure over here, it sums up the process of the entire fermentation process, industrial process, which would include an upstream processing and a downstream processing. So with this slide, we conclude our introductory chapter. And in fact, we are left with just 2 minutes of our Zoom time. So if you have any questions, can I take your questions? Anybody who has questions, they can please go ahead and ask. Okay, then let's discuss about tomorrow. All right. So as you would already know that your class is in the table scheduled to be from at 9 p.m. in the morning, right? So what do you guys say? Okay, there's one more important thing that in like this is the first week and this is the first class. So we are taking it on Zoom, but from next week onwards, I'm going to make your group on Microsoft Team. It is another software, a very nice software which covers this all these classes schedules and a video lecturing platform like we have it in Zoom. So inshallah, from next week onwards, we're going to shift it to Microsoft Teams. So I'm going to prepare your groups. I'm going to share the accounts and everything with you people, and inshallah we can resume on resume it on Microsoft Teams, right? But for tomorrow's class, what do you say? Should we have